filaments that are going to extend over here, over here. We're going to have actin filaments extend here, over to here. So this is one sarcomere. Okay? Now the next sarcomere is also going to have actin filaments attached to the Z disc, but it's the next sarcomere. Does that make sense? So one of the things you might want to do is color this, use a light pink as your actin. And I believe on your model here, if you look at your sarcomeres, anywhere where you have the light pink, that's where you have actin. Can you see on your myofibrils where you have a light pink and a darker pink? Okay. And those black lines that are going, the black lines are basically dividing your sarcomeres from each other.
And those are Z discs?
The Z disc are those black lines that are in the middle of the light pink.
And in between each Z disc is a sarcomere.
Each one of those is a sarcomere. Yes. Okay. So, let's see. Let me use my brown again. Right here in the middle, I have a little M line. This is a set of little filaments that are holding my myosin fibril in place. It's anchoring the myosin. Okay? And instead of dark pink, I'm going to use blue for my myosin. Okay? This is your M line. And my myosin is going to be a big, thick filament here. And it has little paddle-shaped structures that extend up and can attach to my actin filaments. So this is thicker looking, right? This is thicker looking.
So which one is the actin? It's
The actin's a pink. The myosin is going to be the darker or the blue color. Do you see that they do overlap somewhat? They do overlap. So anywhere that they overlap here is a darker band. It's a darker band. It's going to look like that dark pink. So look right here. If you're looking at one of these straws, can you see that you have a dark pink band in the middle with a little short black line in it?
Yes.
That is your M line. And the dark pink is where your actin and myosin are overlapping. Does that make sense?
The dark line is the M line. Oh, dark line is the Z. No. On either side of the Z line is actin.
Yes. You got light pink on either side of the Z line. Do you want me to project this picture up on the overhead? Would that help?
Well, no.
Yes.
Make sure from back here I'm looking at it.
Okay.
Well, yeah, it would
So you're
help.
looking at it like this, but if you lay it down, you can see that these are running the length of the muscle cell, right? And you just have repeating light and dark bands. In the middle of the light pink is your Z disc. So that's this right here. That's in the middle of the light pink band. And then in the middle of the dark pink band is your M line, and that is anchoring your myosin. Okay, so this is myosin. This is your M line, and it's stationary. It's just holding the myosin in place. Okay? And all of this pink or this red color is your actin filaments. And those are going to appear light pink. That and this are all actin. Okay. Now, this area right here where they overlap, that dark pink, is called the A band. So this is called the A band.
So it goes from being a myosin to A band when it overlaps?
Anywhere that they overlap, this place right here, from the ends of the myosin, where they overlap, this is your A band. So it's from here to here. Does that make sense? It's going to be the dark pink. Okay? Now from here over, and from here over, this is where we just have actin, right? This is called the I band. The I band. And I don't know the rationale behind the labeling like this, but this is what it's called. Okay? Now, let me erase this right up here. We know that's a sarcomere. A sarcomere is from one Z disc to the other. Let me put myosin down here. Okay.
We ain't labeled yet is from M disc to M disc.
What's happening? I beg your pardon?
The only thing that we haven't labeled is, there's no labeling from M disc or M line to M line.
There's not something from M line to M line. But there is something from right here to right here. Is there a gap there between the actin filaments?
Yes.
Yes. Now, do you remember us talking about actin and myosin being involved in all types of movement? Do you remember that actin is stationary and that myosin can reach up and pull it? Okay. Those little club-shaped structures on your myosin molecule can reach up and attach to the actin and pull it. And then it can detach and it can stretch back out again and relax. Okay? So, if we get a signal for this little sarcomere to contract, it can contract that much. It can close that gap. Does that make sense? Okay. It can close that gap. So this sarcomere will shorten by about that much, right? Okay. That is called your H zone. And I don't know why it's called the H zone, but it is. This is your H zone. The H zone is the amount that the sarcomere can contract. Basically, it's going to close this gap. Myosin just reaches up and starts pulling the actins, so the whole sarcomere shortens. Does that make sense? Okay. Now, how many sarcomeres are in my myofibril?
Many.
One.
Look at
A
your
lot.
straws. Look
It's
at
a
your
lot.
straws.
Many.
How many?
Many.
Aren't they joined end to end to end? So, can every one of those sarcomeres shorten that much?
Yes.
So overall, the entire muscle can shorten substantially, can't it? Okay? And they will all shorten at the same time, because this message that's delivered by your axon, the signal will go all the way down the length of the muscle fiber, and it will go internal, so that every one of those sarcomeres will contract. Okay? Are you kind of getting a concept of how this is working? Okay. And we do have a number of connective tissue membranes that compartmentalize our muscles. And that's because they're kind of act like insulation. Don't we have insulation on electrical wires? So that the signal is contained, that electrical current is contained. Okay. Now, our first connective tissue membrane is around every one of these muscle cells. Do you see this looks like fabric on the outside of this muscle cell? That is a connective tissue membrane outside of the sarcolemma that's called the endomysium. What does endo mean?
Outside.
Endo means
Within.
inside or within the mesium. Mesium is talking about the muscle tissue, right? Endomesium. Okay. So all of this on the outside of the sarcolemma is your endomesium. Now, if you have a whole bunch of these muscle cells that are in a bundle, that's called a fascicle. So if you have a whole bunch of muscle cells in a bundle, that's called a fascicle. So if I had 50 of these in a big bundle, that's called a fascicle. And then that is covered by another membrane that's called the paramecium. We're going to do this in lecture too, so you're going to get it more than once. So a bundle of muscle fibers is called a fascicle. So maybe 50 or more, 50 or 100, that would be a fascicle. And that is covered by an additional connective tissue membrane that's called the paramecium, which means around the muscle tissue.
Are each of these bundles called something in here?
No,
Okay, good.
no.
Good.
Every one of these is actually a myofibril. So you just have a whole bunch of them inside each of these muscle cells. But we're continuing to organize using these parallel structures, right? So if I have 50 or 100 of these that are in a bundle, it's a fascicle. And then if I have many fascicles that are bundled together, that would form a muscle. Does that make sense? And then around that, we'll have another membrane that's called the epimecium. What does epi mean?
Upon.
Upon, or the outermost. Okay. So every one of your muscles will have epimecium around it, and then between those muscles, you'll have fascia. Remember fascia?
From the rat.
That areolar tissue. So you do have 3 connective tissue membranes that help to organize the muscle. Okay, the first one is around the muscle cell itself, endomesium. And then if you have a whole bunch of muscle cells together, forms a bundle, that's a fascicle. And then if you have many, many fascicles joined together, that will form a muscle. Now that's more than what you had to have for this lab unit. But it doesn't hurt to add a little bit more information so that you understand it better, right? When I first looked at this model, I thought this was a whole muscle. It's not. It's one cell. One cell.
Wow.
Okay? Keep that in mind. This is one cell. It's a really, really, really long cell, but it's made up of myofibrils, which are many, many, many sarcomeres joined together. So it will all work together. This whole muscle cell will contract. Get it organized in your mind. It's hard, isn't it? Okay. Now, she provided y'all that multi-page thing about the organization of muscle. Do you think that helps?
Yes.
Do you?
She
Okay.
usually doesn't do this, but with this particular one, she did
See,
a
she gave me a
YouTube
copy of
thing,
it.
and he draws this on YouTube as your. So we kind of went with that to answer this, and she would just stop it, and it really does help.
Would that be something that you could benefit from watching outside of class, do you think?
You mean the video? Yeah.
Yeah, okay.
Oh yeah.
Y'all gonna put them in the group?
What's the link of it?
He is actually. I wonder how to share that on the the YouTube video of him.
I can make copies of this to give my
You can
class.
just share the URL.
Yeah, you can just share the, but then you don't have anything to write on. You know what I'm saying? So it may be that writing would help be a learning thing. You think you might want that? Okay, she gave me a copy of it, so.
Oh, she did?
Mhm.
Cool. Yeah.
Okay. But so many of the structures look similar to me. So
They
it looks, I mean, the structure that represents the fascicle looks very similar to this. So it starts out by just expanding one of the little fibrils. You have many, many myofibrils that are part of the muscle cell. And then it shows a muscle cell that's part of a fascicle. And then it shows a fascicle that's part of the muscle. You understand what I'm saying? Okay. And the pictures look similar to me. So I don't know how helpful that will be to you. But for those of you that are in my lecture, I'll try to get those copied off for you. And if you want to look at the video outside of class, that will give you more time on it. I really can't afford to use class time looking at a 15-minute video.
So what is the link for the video though?
I'll give it to you.
Okay.
It'll be on the printout. It'll be
Okay.
on the printout. It's a YouTube video. Have you got that on the front?
There's 3 of them in there.
Yeah, Armando, what was his last name? I can't remember.
Okay.
An 8-minute one.
On the top of each of these pages, it has the link for you, okay? So if I print this
Okay.
for you, you'll have the links and you can look at them on your own time. But these pictures all look alike to me.
So she told us the color, that's hers.
Yeah. And you generally will use a pink for or pink or red for your actin because they're thin, and then just use like a purple or a blue for your myosin, and then when they overlap, you know it's got to be darker. So, would you be able to label this picture with everything that I've provided here for you? Okay? Now, just everything that's the Z disc and the M line, make it black, because that's what it looks like on the model, doesn't it? Make it black, and then let your actin be a light pink, and then use something just a little bit darker for your myosin. Does that make sense? Okay. So, and you can see these repeating sarcomeres in your myofibrils. It's a lot of terms, I know, it's a lot of terms, but this will help introduce you to this in our chapters too. Oh, no, it's not this Friday. Okay. Now, in essence, we've covered it all.
Where's he at?
What do you think? We've covered it all. Now, what we can do on this coming Friday is I'll do a real good review and a practice practical, but I'm going to have extra time, won't I? Do you think we might be able to spend a little bit of time on the nervous system in preparation for 5? We can start working on the brain, and then you would do the dissection the following Friday after you take your practical.
I think that's good.
Huh?
Wait, you're just gonna review and then practical and then do the nervous?
No, we're going to do the review next Friday.
Right.
Okay, and the practical will be the following Friday.
Oh, I was thinking the practical next Friday.
No, but what I'm saying is it's not going to take me 3 hours to do the review and the practice practical this coming Friday. Because see, I haven't done this with my classes this week. I've had to do the translations with them because they didn't do them. Okay, let's look at a calendar. Let's look at a calendar. Okay. What is today, the 30th?